Now, as you may know, the PIK program, the PIC program, was created in 2005 to recruit to Penn renowned scholars who hold appointments in 2 or more of Penn's schools. Distinguished by the diversity of their research and teaching, each PIC professor has a unique area of study, but they share a desire to solve complex real-world problems by working across fields in innovative ways. Each PIC professor with us today will consider how our brains shape who we are. And we will approach this through neuroscience, psychology, biology, bioethics, and genetics, as well as machine learning. Our speakers are Shelley Berger, Daniel S. Auck University professor who studies epigenetics and biology, Rob Greist, Andrea Mitchell University professor who studies mathematics and systems engineering, Jonathan Moreno, David and Lynn Silfen University professor. He studies history and bioethics. Michael Platt, James S. Riepe University professor, studies neuroscience and psychology. And Adrian Rain, Richard Perry University professor, studies criminology and psychiatry. And by the way, we will be picking your brains during the Q&A session a bit later, so hopefully you are fully caffeinated or ginkgo bilobed or otherwise ready to participate. So I am delighted to see you all here. Our moderator this afternoon will be Michael Platt. He draws from a wide range of methods in his work, including brain imaging, pharmacology, genetics, and computational science, to understand how and why we make decisions. He holds appointments here in the Perelman School of Medicine, in our School of Arts and Sciences, and in the Wharton School. Once again, welcome, and now please join me in welcoming Michael Platt.
Thanks, Vince. First, I want to thank the Provost's Office and the President's Office for supporting this event, and to President Gutman and Provost Price for their leadership in the PIK program. And of course, special thanks to Sophia Castro, Program Coordinator in the Epigenetics Program, for really organizing this entire thing. So she's been really fantastic. So let's get to the heart of the matter, okay, the brain of the matter. There's 3.5 pounds or so of tissue within your skull. And of course, this is what we're here to talk about. Every thought, every feeling, every sensation you've ever had, every childhood memory, every lover's caress, it's the result of what's going on in this organ. And if that doesn't make studying the brain interesting enough, I think if we take a look at disorders that affect the brain, there's quite an urgency to understand its secrets. So to give 3 examples, 5.4 million Americans living with Alzheimer's disease. If you're a child born into this country, you stand about a 1 in 68 chance of developing an autism spectrum disorder. And about 20% of Americans will experience a mental disorder during some year of their life. The costs of managing and treating these disorders is immense, hundreds of billions of dollars, and lost productivity probably amounts to the same thing. And that's to say nothing about the human toll. So if we could understand the brain in healthy people, if we could understand what falls apart in disease, we could perhaps develop new treatments that would, I think, go a long way to making ours a better society. But the brain has not been easy to crack. It is probably the most complex device in the known universe. So just to give you a few numbers, there are roughly 100 billion neurons making 100 trillion connections with each other, using lots and lots of different chemical messengers between them. And probably there are about 150 to 200 brains in the average human social network. So understanding how brains function is going to be a challenge that is not easily accomplished by a single discipline. And that's why we are here to, in a sense, advertise the ways in which we have applied our knowledge and expertise from multiple disciplines to try to come to a deeper understanding of how the brain works, how it falls apart in certain disorders, how we might fix the brain, and ultimately how all of this knowledge will impact us as a society in terms of ethics and social and legal implications. So I'm going to go ahead and go first. Oh, I should point out first that we will have 5 speakers, including myself. That'll be followed by a panel discussion. We'll also take questions sent in online, and then please join us afterward for a reception outside. Okay, so what I want to talk about is this really fundamental aspect of who we are, which is our capacity and need to connect with others. Okay, so people are almost unique in the degree to which we connect with others. Okay, and we know that this is an important part of the human adaptive toolkit. So the more and deeper your connections, the longer and healthier and more psychologically well-adjusted your life will be. And you may even make more money. Okay, as Clarence the angel wrote to George Bailey in the wonderful holiday classic, It's a Wonderful Life, no man is a failure who has friends. Okay? So this is really a big part of who we are, and over the last 10 years or so, non-invasive brain imaging studies have identified a network of areas that is activated, the so-called social brain network. It's activated when we look at other individuals, when we think about them, when we engage with them, when we feel their pain, when we make decisions that affect other individuals. And in my lab, over the last, say, 10 years or so, sorry. So there are several areas within this network. They all have long scientific names, and you need not remember all those. I just want you to focus on these three things: a temple, a C, and an almond. They stand for the temporoparietal junction, the anterior cingulate cortex, and the amygdala, but you don't really have to worry about that. These are three key players that we have been studying over the last 10 years to try to understand how individual neurons, individual brain cells, actually compute the information that ultimately results in our behavior towards others. And we have been doing this by recording the activity of individual cells in the brains of monkeys who are making choices about the outcomes that other monkeys experience. In particular, what I'll tell you about today are choices one monkey will make about whether to give a reward to another monkey or not to give it to him. Okay? And it turns out that monkeys, like people, are very motivated to give rewards to their friends. They're less likely to give rewards to unfamiliar individuals, and high-ranking monkeys like to give rewards to low-ranking monkeys. Okay, so that sounds pretty familiar in many respects. And what we found so far is that neurons in this temporoparietal junction, the temple, seem to be really important for decoding the current social context. That is, who am I dealing with? What's their state of mind based on the emotional expressions that they are expressing, on their behavior, and how might I think about and predict their subsequent behavior? And we've identified neurons like this one. I'm plotting neural activity as a function of time, and these different traces, you can't quite see it, but they reflect the activity on different trials where the monkey either decided to reward the other guy, reward himself, throw the reward into the trash can. And basically, these are neurons, are about 10% of the population, that just indicate I'm dealing with another individual. Okay, this is a social context, and I'm giving somebody else a reward. And then there is a much larger group of neurons, of brain cells, that actually seem to identify who it is who's going to be the target of my reward. And we have recently discovered that the activity of these neurons seems to be uniquely involved in the predictions, the strategic decision to cooperate with another individual. And I think this is pretty remarkable. We might test that idea by, for example, inactivating this area temporarily and see if monkeys no longer cooperate with each other. Now, the C and the almond, the cingulate and the amygdala, share a really remarkable property in the sense that we have discovered populations of neurons in both of these areas that respond equivalently when the monkey decides to give a reward to another monkey and decides to reward himself. These neurons they're behaving as if they are mirror neurons. For reward, they are effectively calculating, we think, the experience of another individual. I got a reward in terms of one's own experience. What does it feel like to get that reward? And moreover, we have discovered that we can predict the likelihood that a monkey will actually engage in a pro-social decision. He'll give a reward to another monkey just from the activity of these neurons. So the fidelity of signaling within these two areas by these mirroring neurons directly determines whether a monkey is going to engage in a pro-social action or not, be kind to another monkey. Now, these observations raise some very interesting questions. It's quite clear that there's a lot of variation in human social behavior. Some people are kind, some people are not, some people are highly social, some people are not. And at the extremes, there are individuals who suffer from disorders like autism spectrum disorder, schizophrenia, anxiety disorders, who have a lot of trouble making connections with others. And if we could understand what's actually causing those differences, we might be able to remedy them. And the typical way to try to derive understanding in terms of an animal model is to investigate, for example, human genes that are known, for example, to cause autism spectrum disorders, put them into a mouse, and then examine the mouse's behavior and what differences there might be in the brain. But it should be quite apparent, I think, to all of us that the social life of a mouse is quite simple compared to the social life of a human being. And so instead, what we have been doing is going to this wonderful place, affectionately known as Monkey Island. It's an island off the coast of Puerto Rico. It's not a bad gig for any of the undergraduates here if you want to come down and visit over the winter break. And what's beautiful about this island is that there are about 1,600 rhesus macaques free-ranging on the island in about 7 groups. And we have research assistants on the island who are cataloging every single thing that every monkey does all the time. So they're individually recognized and we know everything about them. And we also can collect biological samples from these animals. And we currently have behavioral data on about 800 monkeys, and we have genetic data on about 900 monkeys. And recently, we sequenced the genomes of 225 of these monkeys, selected for differences in social behavior. We discovered about 19 million variants across their genome compared to a reference rhesus macaque genome. And one of the things that was really striking is that in more than about a dozen genes that have been implicated in autism spectrum disorders, we identified a number of variants that had been unknown before. And when we began to examine how those variants might predict social behavior, we found something interesting. So this is basically a map of the social network of a single group of monkeys on the island in 1 year. Think of this as Facebook for monkeys. It's based on grooming behavior, which is their way of kind of having coffee with each other and catching up. And so the monkeys who are in the center have lots of friends, lots of connections. These monkeys on the outskirts have basically 1 or 2 individuals that they can have coffee with. And then, of course, we have this lonely group out here who has absolutely no friends at all. And what's really remarkable is that when we looked at genes that are involved in regulating serotonin function, those individuals who are colored in red in here have basically the double whammy of what you might think of as 2 bad genes involved in regulating serotonin function. So this suggests the possibility that in fact there are genetic origins of these disorders, and in fact we share a lot of the same biology with rhesus macaques, potentially making them a very interesting model for understanding how evolution produces variation in behavior by producing variation in biology. Now, this all leads to the question, of course, of, well, what can we do to potentially change social behavior? And there has been intense interest in one molecule in particular over the last decade or so, and that's oxytocin. Many of you will recognize this molecule as the love hormone, right, or the trust hormone. This peptide is known, for example, to be really important for lots of very basic social behaviors. So, for example, its release is stimulated in mothers at birth and during nursing of infants, suggesting an important role in bonding between mothers and their offspring. And it also, it's also known now to play an important role in regulating attention to others and especially eye contact, okay? And in fact, it turns out that even when you look at your dog, you will have some oxytocin release and your dog will too. So that's a good thing, and actually it's a good way to tune yourself up before you go out so you'll be a nicer person. What's really striking, of course, is that if you know anything about neurodevelopmental disorders, we know that eye contact is one of the first identifying features that predicts the later development of autism in the first year of life for young children. And this is something that many people with autism struggle with. That is, how to just motivate attention, how to be interested in other individuals. And there are no fewer than 36 clinical trials ongoing in this country investigating inhaling oxytocin as a means for promoting attention to other individuals with potential impacts down the line on acquisition of language and other social behaviors. But the real issue here is that we actually have very little understanding of the biology of oxytocin in humans and other primates. We know next to nothing about where it binds. We don't know much about how it affects the brain, and for those reasons, we don't have a good idea about how to dose the oxytocin, really how to optimize it to have the best therapeutic effect with the highest levels of safety. And so, of course, we have been investigating this in our laboratory. Of course, we can acclimate monkeys to, in fact, they seem to enjoy this, to inhale some oxytocin through a pediatric nebulizer. And when we do so, we demonstrated for the first time this actually gets into the brains of these animals. This has never been demonstrated in humans. And when we look at their behavior, for example, in this reward-giving task, this donation task, I'll just ask you to compare this gray line with the red line. The gray line is following inhalation of saline. The red line is following inhalation of oxytocin. And we can see that the monkeys become more and more giving as time rolls on after inhaling the oxytocin. And they also spend a lot more time looking at the other monkeys. So this is very suggestive that this oxytocin is, in fact, having the same kind of effect in monkeys as it seems to be having in humans. And more importantly, we've shown that if we infuse small amounts of oxytocin into the cingulate and the amygdala, those two areas where we had identified those reward mirroring neurons, that we have the same effects on the monkey's behavior. This makes them more giving, this makes them more attentive to each other, suggesting those are the two critical sites for mediating social behavior. So just in the last couple of minutes before wrapping up, I just want to kind of give you a couple of thoughts to chew on before our panel, one of which is that if we thinking about the implications of our discoveries and the discoveries of our colleagues, a lot of the action in the brain seems to be in parts of the brain like the cingulate and the amygdala that we traditionally think of as being important for emotion. Okay, so these are areas that mobilize our emotional responses to others. And they're very sensitive to the cues that other individuals are providing to us, their emotional states. And I think that's very provocative when we think about a lot of things that are going on in the world right now. For example, effective altruism, which is sort of the Silicon Valley approach to charity, which says what we should do is basically calculate where our dollar will have its biggest impact, but you never really see the impact. Okay, this would suggest that you're in fact robbing your social brain of a lot of the information that might actually reinforce giving behavior. And the second is that when so much of our behavior, so many of our social interactions take place online, in the absence of the cues that our brains actually evolved to use, right, to make social decisions, we may be having some very interesting impacts on the processing of social information and really the development of social behavior across human society. So I'll leave those few thoughts for you to take up again in the panel, and now I'm going to turn the stage over to Shelley Berger, who will talk to us about her work. Thanks.
All right. Well, it's great to have such a great audience today. We're really excited about that. And I'll get right into it. Famously, Alice in Wonderland had to choose between a pill to make her larger and a pill to make her smaller. And it may be in the future that you too will be you'll be forced to choose between fundamental changes in your own brain. So the question that I want to pose this afternoon is, would you take a pill to get smarter or to erase unpleasant memories? And so in this classic film, Eternal Sunshine of the Spotless Mind, Jim Carrey had an unpleasant love affair and decided to have what was called a mind wash in the film to rid himself of memories of Kate Winslet. All right, so we work on ants and mice as models for behavior and memory. On the left here, you can see these fascinating leafcutter ants that you see in Central America, and on the right, our clever laboratory mice. So we use the ants for behavioral studies and the mice for memory studies. Now, our research focuses on epigenetics. Epi means on top of, it's Greek for on top of, and genetics refers to DNA and all the genes in your genome, the DNA that makes them up. And so the idea of epigenetics is pretty simple. You began as a union of an egg and a sperm, as you know, into one single cell that typically derives from mom and pop. And in the end, you emerge as a unique individual. Now, what's interesting about you is that the DNA and the genes coming from one single cell are identical in all of the cells in your body. And so your blood cells, your brain cells, your muscle cells, although morphologically completely different, all contain exactly the same DNA and the same genes. And so how do the same DNA and genes give rise to the 200 different cell types that make up our bodies? And so the answer is through epigenetics. So cell identity occurs by switching genes on and off, and this occurs in part through these epigenetic marks. So genes are turned on with these little epigenetic green flags and are turned off by these little epigenetic red flags. And so in your brain, your brain genes have green flags, they're on. Your muscle genes have red flags, and vice versa in muscle, where your muscle genes have green flags and your brain genes have red flags. All right, so we work on ants as a model for behavior. We can't really work on leafcutter ants, they're too complex. Very interesting though. So instead we work on these much more simple carpenter ants that you might see in your garage or in your basement. And this is a typical colony that you would see in my laboratory upstairs here in Smilo, composed of thousands of individual ants. Now if we drill down to the individuals in the ant colony, there are 3 different individuals, 3 different types, the queen and 2 different types of workers, as you can see here. Now, what's really fascinating and what relates so beautifully to epigenetics is that all of these ants are females. They're all females, and even more amazing, they all have exactly the same DNA and the same genes. So much like the cells in our body that divide all the tasks have exactly the same genes and are regulated by epigenetics, so these females are regulated by epigenetic changes. So the DNA, through the epigenetic marks, yield these different animals. Now, I want to drill down a little bit deeper on these females and what they do. So there's one queen per colony, and that queen is responsible for laying all of those eggs, all of the 1,000 of eggs in the colony. And those thousands of eggs then give rise to these 2 different types of workers. The larger, brawny soldiers that protect the colony, and the smaller, brainy foragers that do all the searching for food and bringing it back to the colony. Now, in terms of intelligence then, they really differ quite a lot. The forager has to go out and look around for food, very high intelligence, soldier medium, queen low. Okay, now you may be wondering, what about males? What happened to the males? Well, there are males. They have a very simple task. They supply sperm. That's it. And other than that, their job role is absent. So no comment, guys. I won't go into that any further. Okay, now we're going to drill down on the workers, the soldier and the forager, these 2 very different types of workers. Now, what we wanted to address in the lab is whether we could reprogram the behavior of these brawny soldiers into brainy foragers. And so we can use epigenetic drugs to turn on genes, okay? So we need a simple assay to use in the lab, and so we have these mini nests. The ants live under this red, they can't see red light, and they come out and through this Piking tubing, they exit and go over to this little foraging arena. This is sugar water. This is what they're foraging for. So the very smart one goes out, samples a little, goes back and shares it with her foraging comrades, and they then follow her out, and they're all drinking buddies here out at the sugar water. So that's the assay, and we can take photos of this through time and then use very rigorous quantification to address the foraging. And in this assay, normally, only the foraging ant caste will go for food. The forager, again, is the brainy one. Her genes have green flags, they're on. Her muscle genes are relatively off. And the soldier, the brawny one, her brain genes are relatively off, and her muscle genes and metabolism genes are relatively on. So now we can use epigenetic drugs to alter these flags in the soldier. We can apply the epigenetic drugs to the soldier to alter the flags, to turn on the brain genes, turn them into green flags. And now we have a soldier body and a forager brain. Very smart animal in the soldier body. So we can convert the soldier into a forager. All right, so now we conclude from this that we can make an ant smarter. So the question is, we go back to the question, would you take a pill to get smarter? And I think most of you would likely say no, too many risks to mess around with your brain. Whether that you would do that is an open question. So we can move on to the second question. Would you take a pill to erase unpleasant memories? So now we move to the mouse system where we can carry out rigorous experiments to look at memory. And we use this particular memory test in mice. It's called spatial object memory test. And we introduce the mouse on day 1 into this arena with 3 different objects. And the mouse explores all the objects equally. We can measure that. And now on day 2, we bring back the mouse, this very clever normal mouse, now and move the object. This particular object, we move over to here. And now a normal mouse will now explore this object very will predominantly explore this object and ignore relatively these 2 objects that remain in the same position. So that's what a normal mouse does. Now again, we can use our epigenetic drugs to specifically alter flags to turn off memory genes. And so in the mouse, the normal mouse, we have movement and genes needed for moving around and exploration that are on, memory genes that are on. We now use epigenetic drugs to turn off the memory genes and leaving the movement genes on. So now we introduce our altered mouse back into the arena. On day 1, the altered mouse explores normally all of the objects equally. Its movement genes are just fine. But on day 2, when we bring back the altered mouse, now the mouse is unable to recognize this altered to remember the location of these objects, and so explores all 3 objects exactly the same. So we can erase memory in a mouse. So now we can pose the question, would you take a pill to erase an unpleasant memory? All right, so now let's go back over what we've discussed. Would you take a pill to become smarter? Would you take a pill to forget someone who spurned you? Well, I don't know, probably most of us would be a little bit worried about taking a pill to alter normal behaviors. But now, what if you had a horrible childhood experience? Something that really affected your entire life. Or even more to the point, what if you had a devastating disease of memory, like PTSD? Now we're getting to a really interesting question. So in summary then, our research aims to provide ways to alter behavior and memory, and leaving you in the context of our questions, as in all research, it's up to society to decide how and when to use these therapies. Okay, so I'll leave you with that, and we'll move on. I'm introducing then Robert Greist.
Okay, let's see. Did we tell you about the quiz afterwards? Oh, well, I'm afraid there's going to be math. I hope you're okay with that. This talk is going to be a little bit different. I'm going to talk about the general topic that we're discussing, but from a more mathematical and data science-y point of view. And if we consider not only the problems that we've heard about already, but many of the other problems that researchers on this campus are dealing with, so many of them are related to problems of data, very, very large, very, very complex data. If we're looking at the stuff that's happening in the GRASP lab with controlling large swarms of robots, that is very much a data-centric problem. If we're looking at the work that's done at the Warren Center for Networks, very, very data-centric. if we're looking at the work that's happening in our new nanoscience building, how to assemble very, very small pieces together. Lots and lots of data there. Moving to genetics, we've just heard that's a huge data problem. And finally, the topic for today, neuroscience. These problems are big, these problems are difficult, these problems are new, and very, very new problems require very, very new ideas to attack them, to handle them. And the types of ideas that I work in are mathematical in nature. And mathematics has a lot to offer in terms of how to address these various challenges. Now, I know, right, math. Sometimes the reaction is, well, what's new about mathematics? Mathematics is a bunch of old stuff. We already figured all that out. It's just how hard can you make the calculus problems, right? But there is new mathematics always being generated. Where do we find that new mathematics? What does it look like? Where does it come from? It looks like abstract nonsense, like it always has. Ever since the day when calculus came out, no one understood what Isaac Newton and Leibniz were talking about, and now we teach it to high school students and university freshmen. We build big machines that seem awfully clunky at first, and no one understands quite what they're going to be good for, and then, like ENIAC, they wind up changing the world. Okay, so the area of mathematics that I work in is an area that I like to call the most useful, least used branch of mathematics, in that it has great potential but is currently not being put to much use. That branch of mathematics is a wonderful branch of mathematics is known as topology, whereby topology I mean the study of the qualitative features of abstract spaces. And I'm going to unpack what that means a little bit and then tie it into what we're talking about today. So here's a basic 10,000-foot overview of an area called topological data analysis, where you start off with some data set. Let's say it's in the form of what we might call a point cloud, just a collection of points with perhaps distances between them, or perhaps something much looser, maybe just a notion of proximity. Who is close to whom? Then what one does is, through various mathematical techniques that I'm going to tell you about, we extract qualitative features, maybe holes inside of that data set, and look at how these features evolve as you change the resolution at which you look at things. So what are the details? What's the exact mathematics that winds up getting used to identify these qualitative features? And then how is that useful in data and in neuroscience? So here's where the technical content really starts. I'm going to tell you about something we normally don't tell our undergraduate students about. You usually got to wait till grad school to hear this, so you're in for a treat today. The tool that has been so useful in data is something that's called homology. If you're coming here from a biological background, then you've probably heard that word. It means something totally different in math land. Sorry about that. The problem of language. Homology is an algebraic tool that quantifies the qualitative features in a space. It's something that describes and counts the number and types of holes that a space might have. Now from here, we could launch into 1,000 equations, but I'd much rather just draw some pictures, and you would probably much rather just see some pictures too. So homology comes in a sequence of things, homologies of various dimension. The first is zero-dimensional homology, H0, and what that measures in the context of data is clusters, groupings, pieces. How many different pieces or clusters does a data set break into at the scale you're looking at? That number is measured by this gadget called H0. But as indicated by the suffix, there's more than one. There is H1, which comes after that, which takes a data set and looks for a different type of disconnection or hole. It looks for the type of hole that is like a cycle or a loop inside of a data set, where there's a hole in the middle. Now, 0, 1, what comes after 1? 2. H2 is something that measures a different type of hole, a hollow sort of cavity sitting inside of a data set. And guess what? There are more. Three-dimensional homology, four-dimensional homology, it keeps going, doesn't stop, but it's a little bit harder for me to draw pictures of these higher dimensional holes that you can neither see, smell, taste, or touch, but you can measure them, and you can compute them, and you can put them to use, in neuroscience. Homology is currently having a big impact very recently in this area. I'm going to tell you about some very exciting work that has recently been done in neuroscience. At a large scale, if you take a parcellation of a brain, break it up into different functional regions, and then do an fMRI to see which regions have activities that correlate with one another, you can try to figure out how are the different components of a brain connected together. That's at a large scale. At a small scale, you could go down to the level of individual neurons and probe the electrochemical activity that's happening, look at spike train data over time, and then try to correlate the activity of different neurons and infer from that which neurons are attached or wired up to which other neurons. Both of these are extremely difficult things to do and to get some surety that you've done it right. But when you do it, amazing things can happen. Work back in the 1970s by O'Keefe and his collaborators won a Nobel Prize in medicine 2 years ago for showing that in the visual cortex of rats that have learned a maze, the rats have actually programmed the neurons, wired the certain type of neurons together to remember the shape of the maze. This is intimately involved with how learning happens for these rats, for these mazes. And it's actually giving a geometric or a topological representation of the maze inside of the neural structure. Now that's very different than the way that some other organisms have neurons wired up. For example, if you look at the olfactory system for a fly, it is known, again, through very, very careful experiments, that those neurons are wired up at random. What about in other cases? It's very difficult to do the experiments that can rigorously conclude how neurons are wired together because those axons travel for a very long distance. But recently, there's been some wonderful mathematical breakthroughs involving topological data analysis. If I show you a picture of a network, it's really easy to see the structure. If you can't see what's going on, how do you infer what that network structure is? Here's where homology comes in. We take our friend, the poor rat, stick a bunch of neural probes and test the electrochemical activity of various neurons, do correlations between them, and then as a function of a correlation parameter, we tie those individual neurons together in a sequence of topological objects, simplicial complexes, and we look at their qualitative features as measured by homology. And we use those homologies as a statistic to do data analysis. If we think about increasing the correlation parameter and looking at the size of the homology, the number of holes that you have in your network, then it's known to have the following up-down behavior. So for first homology, number of holes goes up and then it goes down until eventually everything's tied together, no holes. The same thing is true for H2. The same thing is true for H3, but it's known that these peaks come after one another. 1, then 2, then 3. Here's where there's a big difference between what happens in a rat and what happens in a fly. It's the order of the peaks. If you look at a network like a visual cortex of a rat that is geometrically wired, that has learned a maze, these peaks go down in size. One-dimensional homology, two-dimensional homology, three-dimensional homology. For a randomly wired network, theorems show that those peaks go up in size, one-dimensional, two-dimensional, three-dimensional. So now there's a way to take existing experimental data that's not hard to get, run a homology computation, devise a signature, and rigorously conclude how those neurons are wired together, whether geometrically or randomly. updates on this work that was originally done by Chad Giusti, who's a postdoc here at Penn, half in math, half in bioengineering. There have been recent updates from the lab of Danny Bassett here in bioengineering as well. Very, very cool stuff. But it does have one challenge associated with it, that is, how do you do these computations? How do you compute homology? That's where a lot of work in my lab comes in. Some wonderful work by Greg Henselman, who is a finishing up PhD student here in electrical engineering, takes a combination of different classical ideas from mathematics, things that most people have never heard of, like discrete Morse theory and matroid theory, and combining it with some classical matrix factorization techniques to do what was impossible to do a year ago. A year ago, we could only handle systems with maybe 70 neurons. And now, with Greg's improved algorithms and totally new way of thinking about this problem, we're able to handle systems that are much, much, much larger thanks to that work. Okay, so what does the future hold? Well, we're really just at the beginning of being able to figure out how small numbers of neurons fit together. At present, we can do a couple of hundred. What happens in the future when we're able to do very, very large scale and accurate representations of what is happening? What are the implications of that as far as understanding how we learn, how we remember? We're just at the beginning, probably too soon to worry about misuse of such, but nevertheless, it's an exciting time to be working on these things. Thank you.
Good afternoon, ladies and gentlemen. I study the anatomy of violence. So I pick on the criminal brain. And what I'll do in the next 10 minutes is just try and raise and answer two questions. First of all, are criminal brains different to our brains? I hope none of you are criminal here and that they are different. Secondly, what does it mean for society? What does it mean for predicting crime, for preventing crime? And how do we go about punishing crime if criminals have a different brain to the rest of us? First question here, are criminal brains different to ours? We've actually known for a long time that they are. This goes back to a study I did over 22 years ago. Brain scanning 41 normal people, 41 murderers. And the key finding is that the normal individuals have upregulated frontal lobe functioning, part of Michael's social brain. The murderers, however, as a group, lack activation in the prefrontal cortex. Why would that predispose somebody to be violent and criminal? Well, for at least two reasons. First of all, the prefrontal cortex is involved in inhibiting behavior. Secondly, it's involved in emotion regulation. Think of it as the guardian angel on behavior. We all get aggressive and angry at times. What stops us acting out? It's a well-regulated prefrontal cortex that says, no, not the right time here to lash out. But if that guardian angel on behavior is asleep, as it is in murderers, then the devil can come out and people can get killed. So that's one brain region, the prefrontal cortex, part of Michael's social brain. The second part I want to focus on is, again, from Michael, part of the social brain, the amygdala, the emotional amygdala. So this is a study done by one of my graduate students looking at psychopaths. And I think you know that psychopaths are criminals who lack conscience, they lack remorse, they lack shame, they lack guilt. They don't experience feelings in the way that we do. Is that because they have a structurally abnormal amygdala? Jan found, yes, they do, that they have a shrinkage in the amygdala, that psychopaths have an 18% volume reduction in the amygdala compared to normal controls. The blue just indicates the specific voxels which are deformed in psychopaths. But overall, an 18% reduction in part of the social brain. And if the social brain is impaired, Michael, might you expect antisocial behavior to come out? Yes, you would. I want to stick with the amygdala here and talk about moral decision-making because I think it gives another clue about when this area is dysfunctional, then immoral behavior can come out. So we've known from a lot of laboratories that when people have to contemplate moral dilemmas, the amygdala is highly activated. So one example of a dilemma that you'd receive in a scanner is the trolley dilemma. Runaway trolley, it's about to kill 5 railway workers. You are here standing on the bridge. Next to you is a corpulent gentleman. Would you push the big guy off the bridge to stop the train and save 5 lives? 1 for 5. Would you do it? I don't need to know what you would answer. I'm more interested, although nobody did give an answer, did they? I'm actually more interested in how you feel when you do that, when I give you a bit more time. Because for many of us, it feels really uncomfortable, awkward. What do I do? And it's no surprise that the emotional amygdala is activated as part of a wider moral circuit. But what my graduate student, Andrea Glenn, showed is that the higher the psychopathy score, the lower the amygdala activation when contemplating a moral dilemma. So psychopaths, when contemplating these moral dilemmas, that social emotional amygdala is not fired. And to me, this results in or raises a provocative societal question. The Hannibal Lecters of the world, the psychopaths of the world, of course they know it's wrong to kill. But do they have the feeling, the emotional feeling for what is right and wrong? Because I would argue that emotion drives, in part, appropriate moral behavior. And if you lack the feeling for what is wrong, then again, maybe that will predispose you to psychopathic-like behavior. And that raises another question for me. If psychopaths are not responsible for having an amygdala that's, let's say, 3 sizes too small, and if that predisposes them to immoral behavior, then how moral is it of us to punish psychopaths as harshly as we do? And that's where I lead into more the societal issues here. Let's start with prediction. And where does this go? Colleague of mine, Dustin Pardini, we brain scanned men in the community, adult men in the community, looking at their amygdala volume, because a low amygdala volume is a risk factor for antisocial behavior. We then followed the men in the community for 3 to 4 years after the brain scan. And what Dustin and I found is that the blue bars are those men with low amygdala volumes. Their rate of violence in the next 3 to 4 years is 57%, compared to the control group, which is 18%. There's a 3-fold increase in the rate of future violence in men with a smaller amygdala. And that is prediction over and above usual predictors that are made to predict future violence, which is aggression, violence, and psychopathy. So, in the future, would we bring on board brain imaging, possibly molecular genetics, combined with social and psychological factors to do with big data, which Robert was alluding to do a lot better job of predicting in the future who amongst us is more likely to become a violent criminal offender. Prevention, big nut to crack, bad brain, bad behavior, okay? So what do we do to upregulate the bad behavior? And Michael, you could put oxytocin into the amygdala of these psychopaths. I think that would be an interesting idea. We haven't done that, but what my graduate student, Olivia Choi, has been doing is doing transcranial direct stimulation, which is passing an electrical current across the prefrontal cortex to upregulate, in part, the prefrontal cortex. It's a randomized controlled trial where one group get the electrical stimulation to the prefrontal cortex, the other group do not. She then brings them back the next day, and the participants are put in given a social vignette, like, you know, you're in a bar with your girlfriend, this guy's hitting on your girlfriend, you go to the restroom, come back, he's, you know, again, hitting on her, he insults you. Would you whack him over the head with a beer bottle? Would you commit a physical assault? And there's a vignette for a sexual assault. Well, this intrigues me, and again, we've got to be very careful. It's very beginning. It's submitted for publication. But what she finds is that the green group, those who get the electrical stimulation the day before, there's a 47% reduction in their intention to commit a crime. Same thing for sexual assault. Let's be very careful. This is a laboratory study. It's just intent. It's not behavior. But intention is the first step towards criminal behavior. The question is, if this replicates, extends to behavior, would we do it? Would we go into the holy of holies, the brain, to change future crime? Lastly, punishment. Where does this go in the judicial system here? And this is where we get at the integration between neuroethics, neurolaw, and what I call neurocriminology. And this is the question. If these offenders are not really responsible for the brain impairments that predispose them to become criminal and antisocial, what do we do with them? What's our disposition towards them? This is a man who brutally raped and murdered a wonderful young woman. I defended him. He's facing the death penalty. I have him brain scanned in the same brain scanner that I brain scanned those other 41 murderers. I show his brain scan to the court, and I show it compared to 63 normal men who have very good prefrontal functioning. Dante Page doesn't have that. And so I argued that it's brain factors combined with a horrible social environment that this man had that meant he did not really have free will, not as much as me and you have. And he's facing the death penalty. The 3-judge panel bought the biosocial argument that he's just a puppet on a string, pulled by forces beyond his control early on in life, predisposing to this vicious antisocial violent behavior. And he was spared the death penalty. Well, is that the way to go? Let's argue against my position here about early factors beyond the individual's control shaping behavior. Is that just a slippery slope to Armageddon when none of us are really responsible? Because of course there are causes of behavior. Just because we find the cause, should that excuse the behavior? Do we want excuses for heinous crimes like this? And then what does that say about free will? To what extent does that sort of argumentation negate this time immemorial concept of free will and that we are responsible? So yes, criminal brains are different to ours. Where do we go with this? Prediction, would that lead to a Tom Cruise-like Minority Report where you are all brain scanned and those who have abnormal amygdala functioning or prefrontal functioning combined with other factors get locked up before you commit a criminal act, the preemptive strike? And how early do we screen and intervene with young children? Prevention, is it really ethical to go into the Holy of Holies, to change our essence, just our persona? And then lastly, as Portia said in The Merchant of Venice, should mercy season justice? Should our greater understanding of the causes of crime and violence lead us to a more merciful and less retributivist perspective with criminal offenders? And now, ladies and gentlemen, I'm going to introduce to you Jonathan Marino.
So you've already heard some amazing science. I'm going to talk about many of the same ideas and even some of the same technologies in a very different context. So I'm interested in the way that neuroscience figures into national security planning. Now we know that the history of science and the history of armed conflict, and I'm moving from the kind of criminal violence that Adrian works on to organized conflict, war. We know that just about everything that human beings have thought of, come up with, have invented, has been considered as a technology, an approach to fighting wars more efficiently. And in the context of modern neuroscience, let's remember that there are big government projects in neuroscience. The European Brain Project, the Human Brain Project, has as its goal to simulate the human brain. I'm not sure what that means, but I guess like pornography, we know it when we see it. I will come back to that, not the porn part, the simulation part, and not to leave you in suspense. And in the US, the BRAIN Initiative, President Obama's BRAIN Initiative, one of the few things that both parties really like in the last few years, has a more modest goal, but and a lot less money, by the way, than the Human Brain Project in the EU, but still has some very interesting implications. I don't have a lab, or if I have a lab, it's this modest organ up here. But I've been interested in these questions for about 14 or 15 years and published this book with, some of you got the visual pun, which I cannot take credit for. smart artist did that, the brain grenade. And these are some of the topics that I talked about in the book, and this just keeps rolling along. Part of the interest that I have in neuroscience and national security is an idea that's called dual use, which is a term that you've undoubtedly heard. But dual use can come in two different axes, and this needs to be, I think, made very clear. There's the military and civilian axis. There's also the malign and benign axis. And while it's true that a lot of military use could be malign, not all military use is malign. If you're defending innocent people, if you're defending people who can't defend themselves, who are the victims of criminal aggression. And I also want to say that not all civilian uses of a technology are necessarily benign, and I'll come back again to that at the end of the talk. So we know that the national security world in the US is very interested in cognitive neuroscience, and this is just cognitive neuroscience. This is sort of the psychology side of neuroscience. This is some older data that my friend and colleague at Georgia Tech, Margaret Kosal, came up with a number of years ago when she dug through the fiscal year budget in 2011. But we also know that the BRAIN Initiative has as a participant DARPA. The Defense Advanced Research Projects Agency, known to many of you, is the cutting-edge science agency in the Defense Department. And we know that there are some projects that are no longer operating that were part of that period of DARPA, and here's just one, the CT2WS, Non-Invasive Brain-Computer Interface, and these are some of the ideas that were part of that project. If you were lucky enough to get a contract from DARPA to do some of this work, you would know about some of this. And basically, the idea here is to help an operator who's looking at a bunch of video screens be able to record more accurately what potential threats are visible on that screen. Some of you have seen that film about, you're told to, that experiment where you're told to watch the basketball players, and then there's a gorilla walking around, right? It turns out that you didn't notice. Well, we have that experience all the time. So how do you help people who may have our lives at stake because they're looking at some potential terrorist activity through some video screens? How can we make sure that they see these things in a better way, that their perception's improved? And actually, they can use some very old technology. They use some very old technology for some of these experiments. This is an EEG cap. So this is somebody who's just being given some cues. You're not looking where you should be looking, and it gives you a little vibration, a little haptic stimulation, or maybe it just puts an arrow on the screen, so that gradually these operators supposedly got better and better at noticing what they should be looking at. And this is going on all over the country. Here's a very interesting project going on at the Beckman Institute, the University of Illinois. And so here the idea is not to get people to notice a stimulus that they aren't noticing. Here the idea is to enhance fluid intelligence. And fluid intelligence, as many of you know, is the kind of intelligence that helps us get through the day, to be more spontaneous, more creative. Come back to the idea of fluid intelligence in a little while. This gives you an idea of one of the projects that's going on to make us better at managing our lives on a day-to-day basis. And this is just some examples that I pulled off the web of some current projects that are going on under the rubric of what I'm coming to called the third offset. Modeling human behavior, neurocognitive computational models, direct neural interfacing. What's that? Direct neural interfacing. Well, I'm glad you asked. So come back to all this under the context of what people in the Pentagon are now calling the third offset. So what is the third offset? Anybody ever hear of the third offset, by the way? So most of the people that I talk to who are defense planners don't even know what it means, but they're using the term a lot. So the idea of an offset in the national security world is what can give us some advantage over our adversaries. The first offset, if you look at it that way, was the bomb. And that worked for a pretty long time, more or less, mutual assured destruction and so forth during the Cold War. In the 1990s, the second offset was precision-guided munitions, worked very well in the first Gulf War, in Iraq and Kuwait, and still is being used. That technology is still being used, still kind of an advantage, but there's always an arms race. So, people catch up to us. So what's the third offset? The term third offset, if you Google around for it, it's really hard to find it used in a formal way or defined. Secretary Chuck Hagel, Secretary of Defense Chuck Hagel, used it in a speech a few years ago, probably around 2013 when people first started using it. And these are the kinds of ideas that the third offset includes. You notice, if you think about this just for a second, that there's a whole lot of neuroscience going on in here. Every one of these has to do with potentially improving modeling our military systems, our national security systems on what we're learning about the brain from people over here, my smart colleagues. And just this morning, I do read the newspaper at work, by the way. Vince, I'm sorry to tell you I do. The Pentagon, it turns out, according to this New York Times article, put up just this morning at about 11:30, I think. This article describes autonomous weapons, brain-computer interface, fluid intelligence, does not mention the phrase the third offset, I'm a little disappointed. But so much of what is going on in this article, just went up this morning on the Times website, your homework is to go read it, is part of what I'm talking about to you the next few minutes. So we know that you can do brain-to-brain interface. This is an experiment that was done about 4 years ago at the University of Washington in Seattle. EEG cap, TD, transcranial direct current stimulation, sending an impulse. In a way, this is very cutting edge. In a way, it's kind of primitive. This is not an efficient way to get people to talk to each other brain to brain. And one of the problems is that the materials we have, the technologies we have to do this are pretty simple. So this is what's known as the Utah array, 96 microelectrodes. That's pretty much the chip that gets put in people's brains for various experiments. It's the kind that's used in experiments with people who have tetraplegia. You've seen these, I'm sure. They learn how to move a cursor around a computer screen, control the lights in a room, control the temperature, and so forth. Pretty primitive. There's one company that now is marketing 128 microelectrode array, still not much. What DARPA has in mind, which is described in this little press release, is, and they don't use this phrase, but I've talked to people there who will tell you that this is what they have in mind, pun intended, a microelectrode array with hundreds of thousands of microelectrodes, hundreds of thousands, maybe 1 million. What could you do if you put one of those 1 million microelectrode array in my brain at just the right place and one in your brain? We have no idea what that could mean. But it might mean that we would be able to talk to each other directly. So I wouldn't have to say, hey, look over there. I would say, well, you would just know what I'm thinking. And you would be able to respond to it instantaneously. This also gives us the possibility we could network a lot of brains together. So somebody I talked to at DARPA said, you know, wouldn't it be really cool if we could get all these amazing, smart people we have in this country and confront them with a military problem, a tactical problem, but network their brains together so that they could all be thinking simultaneously, be using all that fantastic material they've got in their heads, all that knowledge, all that experience, all that wisdom, and all that expertise, all that education, and use it together at the same moment. Wouldn't that be great? We could solve a lot of problems that way if we could network our brains instantaneously. In fact, it's already being done. I don't know how they do this. You got to ask my colleagues. Maybe Shelley understands this. Maybe Mike understands this, or Adrian. I don't know. But they are somehow getting monkeys to network their brains so that they can solve a problem. And so we're kind of moving in that interesting direction. Here's another one that I want to kind of begin to wrap up with. Here's a problem that the Office of Naval Research is working on. I just want you to look at this phrase. The goal is the development of biological neural networks, et cetera, et cetera. You don't even need to go there. The development of biological neural network. I thought that's what I did when I was growing up. I was developing a biological neural network. Well, what if we can develop our own biological neural networks now using the smart people that we have in front of the room here in their labs or in their students' labs in 10, 20, 30 years? What would that look like? What kind of creatures would we create? Well, we might create a creature very much like the ones that fascinate me. I'm not getting, I don't get 10% or anything for showing you this ad. In this new series called Westworld. And I'll just point out to you that this is not a new trope in our history. 2 years from now, we're going to be celebrating the 200th anniversary of a little novel that was written by a brilliant young woman named Mary Shelley on a long rainy weekend in Geneva, Switzerland. It was called Frankenstein. See you in the future.
Ask my colleagues to come up to the front, and we'll have some time for questions. And Sophia, I think, has a laptop, so we can get some questions from the internet as well. Thanks. So we'll go ahead and open up the floor to anyone who has a question to ask for any of the speakers today. Back there in the back. Do Do we have a microphone run up there? Yeah, thank you.
So this question might cut across more than one speaker, and that is, in the examples we saw of neuroimaging studies, I think all of them were done among individuals who were already mature, had developed whatever psychopathic or other characteristics that provided the reason you were studying them. And so my question I'd like the panel to address is, what are the limits in studying people's brains who have already developed the characteristics that you're interested in, especially in terms of chicken and egg?
Okay, chicken and egg question is a very important one because, of course, brain imaging is correlational. The best I can give you on that is a couple of things. First of all, we've been brain scanning 11-year-old kids here in Philadelphia, and they're not psychopaths, but they have what are called callous and unemotional traits. And that's as described by the parents. It's a forerunner to psychopathy. And even at age 11, it's a sample of 300, which is relatively bigger for brain imaging. We do again find the structural shrinkage in the amygdala in these callous and unemotional children. You can't do brain imaging on really young kids because they move around in scanners. You can do it on babies. Nobody's done a follow-up study from, soon after birth to later on. But what we have done is we've taken an indirect marker of amygdala functioning. It's called fear conditioning. We've measured that in 1,800 children and shown that kids with poor fear conditioning at age 3, suggestive of poor amygdala functioning, they are more likely to grow up to become criminal offenders 20 years later. So the chicken and egg question, we think the brain impairments precede the abnormal behavior, but could it be that living a violent way of life could impair your prefrontal cortex? Absolutely, because they have accidents, injuries, violence offenders are risk takers, and there could be external damage to the prefrontal cortex, for example, from head injuries, so that the violence does precede the prefrontal dysfunction.
Jerry.
Two related questions. Have there been any studies on the link between the amygdala and terrorist behavior? And are there easy way of detecting it? So is the next wave of detection in airports and others is to try to identify the amygdala?
Do you want to go?
Well, that was, there was a little remarked upon conference in 2010 that I attended at the NIH that was sponsored by various, like Stratcom and the North American Command and so forth. It was a Pentagon, largely Pentagon and DARPA sponsored, but it took place at the NIH campus. And the topic was, is it possible to put together some of the information that we're getting from neurobiology and to predict political violence? So there is interest in the questions like this, and it would not only be sort of anatomy specific, like the amygdala, but also what's going on in the environment that triggers large social networks to operate in certain in violent ways and so forth. It's obviously incredibly speculative. It's very controversial stuff, but there is sort of an abstract long-term interest in that question. The other question, the people were laughing about it, but could you detect somebody's violent ideation in a crowd? Well, you can't really use the MRI, right? Because people are wearing a lot of metal will pull them into the magnet. So people have asked that question, and I've been on a couple of National Academies committees where that question has been asked, but the technology just doesn't allow for it now. And nonetheless, there are a lot of people out there, I can tell you, who believe that their brains are being scanned all the time by some malign entity. And I tell you, if you write a book called Mind Wars, it ain't so funny. There are many people who have this view.
I'll just add very quickly, there's no research on terrorists, so it has to be, as Jonathan says, speculation. One does wonder, however, whether terrorists are fearless individuals, that they are risk takers, and we know the amygdala is central to fear, And one wonders whether they would show reduced amygdala functioning to threatening stimuli, for example, in a scanner. But, again, there's no marker for terrorism. There's no marker for crime, violence. It's a complex mix of lots of factors. And frankly, I've really oversimplified a very complex issue here.
Mm. And also, some people who get caught up in terrorist activities are themselves victims, right? They're given drugs or they're put in impossible situations. So it's very hard even to know what your subject population would be, even if you could look at them.
Jerry, did you have a follow-up or not? No. Okay, John?
So what was occurring to me was when you were, oh, is there a different John?
No, you.
I'm suggesting that she bring
There's
the
only one
microphone
John.
down so everyone can hear you. She's going to bring it down.
What was occurring to me was when I was looking at, is anything coming out of this thing?
Yeah.
I think so.
When Michael was showing his hierarchy of social interactions, and I was thinking of Shelley's ability to use epigenetics, or the potential to use epigenetics, you can't help but wonder, we know that these hierarchy functions are based on hormonal differences, which are going to lead to epigenetic changes. And I was wondering, is there any ability or thought, even in lower animal forms, where you take predispositions, risk factors, such as you found, Michael, and use epigenetics to overcome these risk factors or these impoverished outcomes, impoverished behavioral outcomes?
All right, I'll just say that we're starting to work together to address some of these questions with his animals, whether we can profile the epigenetics in the social isolated animals compared to what he calls popular animals, see if there are epigenetic differences by doing profiling, because he now has a brain bank of these animals. And then we can address whether we could actually try to alter behavior using epigenetic therapies in social situations. I think that's what you were asking. And I don't know, Michael, do you want to comment?
Yeah, well, I mean, I think it allows us to at least get closer to kind of cracking this chicken or egg problem, which is where we have basically the full genome sequence of every single animal. We have a deep behavioral profile. We know everything about the environment in which they grow up, and we know everything about their parentage and their ancestry and going back. And we will now have data on gene expression in these core networks that are involved in social behavior, and ultimately we should be able to unravel this. I mean, it's going to be complicated. We're going to need Robert's math, probably a lot of
Right,
it,
I was thinking that.
to make sense of that. But
Yep.
in principle, this should work.
Actually, if I can just follow about listening to Robert, those of you from an epigenetics background know that we are getting to single cell and single molecule epigenetics. So I don't think it's far-fetched in the not too distant future that we can look at the topology in the brain in spatial. I talked about spatial recognition, and that's what Robert talked about. In fact, that's how the whole thing started. We got talking in an airport lounge 2 years ago and realized we were both interested in a similar problem. So it's not too far-fetched to think that we could look at look at the lighting up of these cells and then pluck them out, of a mouse or whatever, research organism, and now do the epigenetic profiling and see what's happening in real time. And Robert, you want to say something?
Well, one of the assumptions that I find a lot of people have is that if we just wait long enough, the computers will get fast and then all the problems will be solved. But so many of the problems that we're dealing with in genetics, problems of protein folding, all kinds of things, we need better than Moore's law in order to be able to address them. And if you look over history at a competition between the speed up in computers versus the speed up in the algorithms that are used, the theoretical advances, the human Moore's law often beats Moore's law for semiconductors.
Great. Another question?
Hi, it's such a pleasure being here among the famous brains of University of Pennsylvania. You all do so much to contribute to knowledge. But there are certain broader issues. In philosophy of mind, for instance, there's the issue of is the mind and the brain the same? And there's a difference of opinion there. Secondly, the point which Adrian made, that this is primarily a correlation and not causation. Also, Adrian had introduced the idea of free will, that is seeing the brain scan of the murderer, the murderer doesn't have free will. So the question is, what is free will? And to understand free will, we need to understand what the nature of freedom. That freedom needs a free society to be possible. But that's where the conundrum is. On the one hand, society cannot exist without caring for the other. But if you are free, why should you care for the other? So how do you have a free society and free will?
So maybe, well, anyway,
do you have a free society?
Tackle that.
I'll volunteer on this one. So if you look at those of us who are up here, we span a whole bunch of different disciplines, a bunch of different colleges here at the University of Pennsylvania, but we're just 5 people. We're not spanning everything. And so many of those questions that you raise, there is so much to be offered from our colleagues in the humanities about what it means to be human, what it means to have free will, what it means to be more than just a collection of electrochemical reactions going on in the brain. And that is part of what the larger Penn community helps us answer and address. And just because there's no one representative of that here this afternoon, that doesn't mean that that is not vital to answering these questions.
This is why I love Rob, because how many mathematicians would ask these big philosophical questions?
I think in the interest of inclusiveness, we'll ask us for some other questions.
I'm going to take a stab at the philosophy angle, though.
Oh, I was wanting to do that
I
too,
could.
but you go on.
Well, okay, you can too.
No.
So much smarter with that British accent about these philosophical questions. Anyway, I'm jealous. So, on these questions about mind, brain, and free will and determinism, my favorite philosopher is William James, who said, we don't really solve philosophical problems, we get over them. And I think what is going to happen with much of the work that my colleagues are doing is that we're going to reframe these questions. In 50 years and 100 years, they'll look very different.
Hey guys, thanks for speaking today. It was very interesting to hear. My question is whether or not it's right to make some of the changes that we talk about. Let's say we have one of these young children in Philadelphia that's expressing signs of pre-pathology, and we have this electro treatment that we can use to stimulate the size of the amygdala, et cetera. At what point, how can we draw the line between pathology and a component of personality? Like many of you may be familiar with the idea that folks with Asperger's might contribute in a great way to mathematical computation or something like this. So where do you draw the line between pathology and personality, and when is it right to change somebody's future personality in the hope of avoiding a future pathology?
I'll answer the first
Wait
part,
one second,
but not the second.
before you do. Riley, who's a graduate student in my lab, did the ant experiment. So his thoughtful question comes from the guy who did the ant. So congratulations to him.
Well, anyone else first?
No, you take it.
Start there.
You go ahead.
Okay, on the first part of the question, it's a conceptual approach. I take a dimensional approach to psychopathology. I think in most cases, it's not black and white. It's many different shades of gray. Not necessarily 40, but a number of shades of gray. It's a dimension. Dante Page, the murderer there, I would say he had some degree of free will, but it was greatly constrained by factors beyond his control quite early on in his life. And I haven't given you the full detail there. But it's shades of grey, more or less, not dichotomies, with some exceptions. The second question is much more difficult to deal with. I could have a stab of it, but does anyone else want to have a stab? Shelley, you talked about, should we go into the brain? And you asked this question too. I was really following on from you. Is it right to meddle and change our memories, change our being? Is it right for us to change human nature, the essence of it, our brains, to change, albeit for the good, to make a safer society? But is it changing them irrevocably?
Well, that's why I posed it that we might not want to do it if it were a matter of changing our personalities. I think that my answer would be no in general. I think society would want
It's a
to.
societal, it's a personal issue too, and the fact of the
It
matter is
is.
people are doing this anyway.
Well,
on their
it's
own,
true.
Because a
I
lot
mean-
of these technologies, especially brain stimulation,
TDCS,
transcranial direct current stimulation
Right.
is essentially direct to consumer. People are doing this, and they're actually so that they're reporting on their experiences. So it's I think it's going to happen, and so we need to be prepared and be very thoughtful about what the implications are going to be.
Just as a quick comment, I mean, our society's already decided, right, that we can change our brains, that we can relieve depression or
Yeah,
anxiety.
that's true.
So we've really crossed that sort of Rubicon. We've really decided that in some cases, under some circumstances, that we will change the brain. Now, those are reversible technologies, and that may be one of the things that really gives one more pause.
Right.
You know, if you make a lesion or do something that's not reversible, and you say, I don't really want to do that, well, then it's much more difficult. But it seems that we've decided in some domains that it's okay to change the brain and that we want to, that we don't think.
But
there's
there
something
are,
noble about suffering
there
like
is
that.
also the potential for unintended consequences. And so I had a slide up of John Elder Robison's book, Look Me in the Eye. And so he's a well-known, very famous man with Asperger's syndrome, and he had has had this wonderful, successful life with an early career doing explosions for the rock band Kiss, and he's worked for the Defense Department. He's done all kinds of amazing things. He didn't really discover he had Asperger's until he was in his 40s, and he recently had begun undergoing transcranial magnetic stimulation to improve social function. And what he found was that, in fact, suddenly he was awash in incredible sensitivity to all of the lives of everyone around him. And it was sort of wonderful on the one hand, but it led to the complete dissolution of his marriage, and he got divorced. And so then you have to, ask again. He asks the question, what would I have done that again? To have the experience of being able to connect with people, but really then not being able to really connect in the same way I had with my spouse.
So just to add to this really good question, there's also a political philosophy angle, namely, but we were talking about individualized uses and sort of self-determined uses, DIY, but should a state have the authority to make these changes, a kind of different slant on it. I must say on the mental illness and creativity angle, I'm probably the only person in the room who grew up in a mental hospital. Not as a patient. My father had a small mental hospital in upstate New York. And I knew lots of people who had schizophrenia, and I can tell you they were not creative. They were not, and they were miserable. So I'm a little old-fashioned about this. I think in general, I think a true psychopathology makes you miserable and makes you unable to be creative and to become who you would like to be.
I think I'd just add on this, that I don't think any psychopathic child or any serial killer as a boy grows up wanting to be a serial killer. Boys want to grow up to become great baseball players, football stars. Yeah. And I think there are factors beyond their control, even starting before they're born. Mothers who smoke during pregnancy, that raises violence in the offspring threefold, correcting for all background factors. These fetuses are not responsible for that. We're not blaming crime on women per se, but there are very early factors beyond the individual's control that at least raises the odds of future offending.
So maybe if we had the tools and we could measure and tell folks that they had a risk, and then
they could choose
to decide whether or not, just like we use drugs today, they want to have one.
And the question I think that Jonathan's raising is the big brother question, whether the decision-making goes beyond the individual and their family, and it goes to government.
There's a question over here.
Are there any implications of this research for political decision-making, why certain political candidates appeal to some individuals?
Well, the Times, on a couple of occasions in previous election cycles, ran functional MRI images of people looking at pictures of Hillary Clinton or Barack Obama and so forth. You haven't seen that this time, and I wonder why that is.
Well, there was a backlash by the neuroscience community was
really upset about it.
Pretty fierce because that was a fairly simplistic example of the
I'd still like to see it this time.
So, I mean, where basically because you see some area light up, you interpret it, because that area lights up in,
Right.
in other kinds of occasions, that is what that person was thinking, which is not necessarily the correct inference to make. And so I think that's why you haven't.
We don't need an fMRI this time for
No.
insanity anyway, do we? We can tell.
Right.
Something's going on.
So I think we have time for one more question before we wrap up and head out to the reception. There's a question up here.
Hi, thanks for all the great talks. I was just wondering if each of you could maybe give a little comment on how you might apply the things you're learning in your research to societal problems such as the opioid epidemic, which is what I've been thinking about a lot. So,
Anyone want to take a crack at that?
The addictive brain? Is there such a thing?
Yes. I mean, I think that, addiction is really a pernicious problem, and you have essentially chemicals that are introduced into the brain that bind to and are hijacking the systems that, would normally be serving a function that's adaptive. And so I think the question is really about how do you prevent individuals from beginning in the first place, and that seems to be in the case of the opioid epidemic, more of a social problem, right, than potentially a brain problem. Once people have become addicted to opioids, then I think that's a different question. How do you get them off of that? But if we could prevent the sort of dissolution of social connections, and essentially, like I was talking about, build stronger connections between people, then maybe we could avoid the temptation and the feelings of relief that one gets from the sort of standard anxieties that individuals are feeling. So with that, I think we'll wrap up. Thanks all of my great colleagues. This has been a great trip to be on, and thank all of you.
Thank all of you.